This video is about the mechanism of color blindness. It is the second in our series about color blindness. Color blindness is surprisingly common, occurring in about 1 in 8 men, but only 1 in 50 women. Most don't even know they have it unless they are tested. It can affect day-to-day functioning to various degrees, and sometimes even career choice. We begin our story with John Dalton, who is known both for the development of basic atomic theory and is one of the most famous examples of color blindness. His description of how he saw colors gives us a particularly good insight into the concept of color blindness. Here is the color spectrum as someone with normal color vision sees it. Dalton, looking at the same spectrum of colors, described what he saw. Quote, "I found that persons in general distinguish 6 kinds of color in the solar image, namely red, orange, yellow, green, blue, and purple. I see only 2 or at most 3 distinctions. These I should call yellow and blue, or yellow, blue, and purple. My yellow comprehends the red, orange, yellow, and green of others, and my blue and purple coincides with theirs." End quote. From that description, a non-colorblind person can get a pretty good idea of what the world looks like to someone with his particular kind of color blindness. As a scientist, Dalton was curious. What was different about his eye? He guessed it was because the fluids inside his eye were tinted blue. He was curious enough that he directed that his eye be examined after his death in 1844. The fluids and media were found to be clear, not blue. There's more to the story. Some of his eye tissues were preserved so that in 1995, when the technology was available, gene testing was done. It showed that the green, or M-cone pigment, was absent. That put him in the category of red-green color blindness. In technical terms, because he used 2 color cones, that made him a Dichromats, and more specifically, missing the green cone made him a Deuter-anope. We will explain the terms later on. This was such a famous example that color blindness often went by the name Daltonism. As humans, the rich level of color vision that most of us enjoy is based on having 3 color cones, resulting in what is called trichromatic color vision. We also have rods for vision in low light, but they don't contribute to color vision. Things, of course, don't always go as planned. Color deficiency usually occurs in one of 2 levels. The simplest to understand is when one entire cone class is missing. Actually, it's possible to miss more than 1 cone class, but that is rare. For example, if the green cones are missing, as we saw with Mr. Dalton, in humans this results in a deficit in red-green color vision. You might find it interesting to know that for most terrestrial mammals, this is their standard level of color vision. The second level of color deficiency is more subtle. There are 3 cones, but one of them is a hybrid or chimeric cone. This is a bit more complicated, so we'll take more time with it. Some years ago, the theory was that all the cone classes were present, but one of the cone pigments was not functioning properly, so perception of that particular color was just weak. This shows the concept of a weakly functioning green cone. A good deal of research starting in the 1980s revealed a more complex explanation. Instead of having a partially functioning cone, I am now showing a hybrid or chimeric cone pigment. I'm exaggerating a bit with the yellow color to make the point. It actually looks more like this. One version comes with a red and red-like cone, and the other version a green and a green-like cone. We will return to those later, but first, let's figure out how we got there. Here are all 23 pairs of human chromosomes. There are 22 pairs of autosomes, and the 1 pair of sex chromosomes, the X and Y. The gene for the short wavelength or blue pigment is on chromosome 7. The gene for the long wavelength or red pigment is on the X chromosome. The X chromosome is also the location of the gene for the middle wavelength green pigment. Taking a closer look at the X chromosome, the L and the M genes are actually located right next to one another at the end of the long arm of the X chromosome. So close they share the same address, XQ28. Being on the X chromosome and in close proximity has two consequences, which we talked about in the introductory video. First is X-linked inheritance. Second is that crossing over or recombination frequently occurs between the two genes. What does that mean? When a cell divides, the chromosomes and the genes they carry are passed on to the daughter cells. In the process of cell division, chromosomes gather in the center of the cell with like chromosomes pairing up. As the single parent cell splits into two daughter cells, the chromosomes are supposed to be divided up equally. It may surprise you to learn that frequently the duplication and distribution of genes is not as neat as, say, dealing cards. An example will help make this clear. Here we are showing one maternal and one paternal chromosome aligned together at cell division. I am showing two genes on each chromosome coded by letter and color. A single gene may come in different forms, which are called alleles. I am showing gene 1 with alleles A and B, and gene 2 with alleles M and N. Each allele is a different form of the same gene that could code for different blood types or different opsin pigments. At this point in cell division, the chromosomes do a surprising thing. They may exchange segments. This is called crossing over and results in the recombining of the genes. Each daughter cell then gets one of these chromosomes, but with a different combination of genes compared to the parent. It is also possible that something else might happen at crossover. Here is a pair of X chromosomes showing the genes for red and green pigments. Here they are crossing over. If the crossover event is not equal, then one possible result could be as shown, with whole genes gained or lost. Remember that males have only one X chromosome. A male who gets the chromosome with one red and two green genes should have normal color vision. The other chromosome has only a red gene. A male who gets this chromosome would have a form of red-green color blindness called Deuter-anope, like Mr. Dalton. A daughter who gets this chromosome would be a carrier for Deuter-anope. If you think unequal crossover is unlikely, the modern ability to do gene sequencing has turned up the following result. On the human X chromosome, the expected complement of one green and one red occurs in 25% of people. 50% have one red and two green genes, and the remaining 25% have 4 or more genes. How about that? And if that isn't enough, the genes don't necessarily have to be exchanged as a whole. It is quite possible crossing over will break them in the middle. Here the arrow is pointing to a hybrid or chimeric gene. On this chromosome, the gene for the long wavelength pigment is normal. But the first gene for the middle wavelength pigment is a hybrid. That will affect the range of light it is sensitive to. The second arrow shows the single hybrid gene on the other X chromosome. This is time to pause and realize we have arrived at an interesting place. Recombination involving the red and green genes has resulted in chimeric genes throughout the population, the results being considerable variation in sensitivity ranges to red and green colors. The proximity of the red and green genes on the X chromosome is why these two colors are linked together in the most common form of color blindness, red-green. Let's look at that in more detail. Because this is important, I'm going to take a little extra time to repeat the last couple of examples using a different diagram. The red and green arrows represent the genes for the respective pigments. This is the order as they sit next to each other on the X chromosome, and there may be multiple copies, as we discussed. Even though there are multiple copies of the green gene, usually only the red and the first green gene are expressed. Remember that, it is important. Case 1, the simple case. A recombination event occurs that results in a green gene being shifted from one chromosome to the other. A male offspring who gets only the red gene is a Deuter-anope, like we talked about with Dalton. If he gets the combination of red plus the extra green, then color vision is normal. Case 2 involves recombination with splitting of the genes. That results with a hybrid red-green or green-red gene going to each daughter cell. The final color behavior of the hybrid gene depends on where the gene was split. In this example, we will say the upper hybrid behaves as a green-like pigment. Then the result is the same as lack of a red pigment, or protanopia. The other descendant would then have a combination that had a red and a red-like pigment. That result would be some degree of red-green defect. Remember, red and red-like, and green-like. Researchers have spent a lot of time characterizing different hybrid genes. Here is an example of multiple possible substitution sites and their spectral sensitivities. The opsin pigment contains 6 segments, some of which are more important in determining color sensitivity. The particular version of segment 5 determines most of the color behavior. Here are 2 examples of hybrid genes after crossing over. The upper pigment, with segment 5 for green, will behave as a green-like pigment. The lower pigment, with segment 5 for red, will behave as a red-like pigment. There are more ways the genes can be split, but I'm not going to list them all here. You can find those in the references. In the resulting opsin pigment, the spectrum and its peak would be shifted by a variable amount. For example, here I am showing 2 of the several possible ranges of the sensitivity spectrum for the green-like pigment. And here are some of the several spectrum ranges for the red-like pigment. The main concept is that with gene recombination, the peaks in cone sensitivity are variable, which is the basis for variability in red and green sensitivity and the red-green forms of color blindness, which are the most common. We will finish by reviewing the technical terms for the different levels of color deficiency, which will serve to help organize and review the subject. The first level of color deficiency occurs if you have less than the full number of cones. A monochromat has only 1 cone and sees the world in grayscale. A dichromat sees the world with 2 color cones. That gives them the minimum ability necessary to distinguish between colors. A trichromat sees the world with 3 color cones. This results in the richer color palette that we see. It is not necessary to stop with just 3 cones. A number of animal species, and possibly some humans, can be tetrachromats, but I don't have the color palette to illustrate that. The next set of names gets a step more specific about which color is missing. These people all have 2 functioning cones, so they are all dichromats. If someone is missing the red cone, they are called a protanope. If someone is missing the green cone, they are called a Deuter-anope, and if they're missing the blue cone, they are a tritanope. The last level of color deficiency names describe those who have partial color vision defects. They have 3 cone types, so they are trichromats, but not fully functioning, so the term is anomalous trichromat. It is important to note the level of color deficiency varies from mild, near normal, to significant, like a dichromat. Having blue, green, and a green-like pigment is called protanomaly. Having blue, red, and a red-like pigment is called Deuter-anomaly. Here again are the cones, blue, green, and green-like, protanomaly. Here's what that looks like on the cone spectrum. The cones blue, red, and red-like is Deuter-anomaly, and its cone spectrum. The blue gene on chromosome 7 does not form a hybrid like the green and red ones do. Rather, it undergoes one of several mutations that can reduce its sensitivity. It is affected much less often than the green and red pigments. Here is a simulation of what the spectrum looks like with the three different kinds of color vision deficiency. The protan and deutan fall into the category of red-green deficiency, and the vision result is quite similar. The tritan falls into the category of blue-yellow deficiency, and the result is quite different. Here is a chart showing how often it happens that one cone class is missing. These are the dichromats. You can see that in males, missing a red or a green cone occurs about 1% of the time. Missing a blue cone is much less common. This chart shows the trichromats, those with two normal cones and a hybrid red or green cone, or a defective blue cone. Again, in males, the red-green deficit is much more common. Here is where we are in the series. In the first video, we presented a review of color vision and basic genetics as background. That included location of the colored genes on different chromosomes, X-linked inheritance, and recombination of genes. In this video, we built on that information to show how inherited color deficiency happens, either by missing a cone or by having a hybrid cone. In the next video, we will use what we know from color science to understand color deficiency in a more technical way. In the last video, we will take a descriptive approach, simulating what the world looks like with color deficiency. If you want to read in more detail, you can go to the following selected references.